All right,
Hello.
well, I think we'll go ahead and get started. Thank you all for joining us. I believe this is our sixth gathering of Forest Talk, where we talk about the plants and the animals and all the cool things we find in our native Ohia forests. Today, we welcome Dr. Natalie Graham from the University of Hawaii-Hilo as our guest speaker. You've probably heard the term arachnophobia. Well, today we're here to talk about arachnophilia, why you should love Hawaiian spiders this Halloween and all year round. Just admitting a couple people. Hawaiian spiders are incredibly diverse and full of surprises, from colorful leaf dwellers to web building specialists found only on single mountain tops. This talk will explore how these unique arachnids reveal patterns of evolution, ecology, and biodiversity across the Hawaiian archipelago. We chose this date, October 29th, for a very special reason, which I'm sure you've all put together. Halloween is coming up, and maybe this talk will inspire you to be a spider convert, as I am. And bonus, it might give you some special, one-of-a-kind Halloween costume ideas. For those of you who might be some of my forest journalers, I invite you to get your journal out and start a study page on Hawaii spiders. My name is Kim Rogers, and I'm an outreach specialist at Kauai Invasive Species Committee. My focus is ohia and protecting our native forest ecosystems from rapid ohia death and other forest threats. We have a multi-agency collaboration, many partners who work with us and make regular surveys of our ohia forests to monitor for you the health of Kauai's watersheds. And as you've probably heard me say before, a healthy ohia forest also helps ensure a healthy ocean reef. So before I introduce Natalie, just a couple of announcements. If you're just joining us, please drop in the chat where you're joining us from, and if you're affiliated with an agency or an organization. As always, please follow good Zoom etiquette and mute yourself. If you do have questions, we welcome them. However, if you would drop them in the Q&A, which you should see on the bottom of your screen, an icon for that, not in the chat. That just helps me find the questions in case the chat gets chatty, which I hope it does. We are recording this and we will send out the recording probably by the end of this week, as well as we'll upload it to our KISK YouTube page. That's where all of the 5 previous Forest Talk sessions are stored and archived. I'll drop the link in the chat uh, so you can find those. Uh, we've talked about our native land snails. We've talked about our native forest birds, our native and endangered seabirds, lots of great forest dwellers. Um, I do have to make an announcement about our planned November Forest Talk. That was supposed to be about our fancy case caterpillars in Hawaii. Unfortunately, due to the government shutdown, we've had to reschedule that until hopefully, fingers crossed, it will happen in January. So, but we do have one more Forest Talk scheduled for this year, and that will be December 2nd with Mike D'Amata, a man with a generous amount of native plant knowledge. I know he's well known around this island at least. You can watch our newsletter for the registration link that will be coming out in the next week or so. And if you're not on our newsletter mailing list, I am going to drop it in the chat too in a minute once I turn things over to Natalie. One other announcement, and that is hopefully you have on your calendar Saturday, November 8th is Ohia Love Fest. This is an annual celebration of all things Ohia. And as usual, it will be held at Limahuli Garden and Preserve starting at 10 a.m. It is absolutely one of my favorite events of the year, and it takes place in a most amazing location. So I hope you can join us for that. I'll drop the link for that, more information in the chat too, in a minute. But now let's talk spiders. Dr. Natalie Graham is a molecular ecologist and assistant professor of biology at the University of Hawaii at Hilo, where she leads the Elala Biodiversity Lab. Her research explores how human activity reshapes Hawaiian ecosystems, and she does it using DNA from specimens and environmental samples to uncover patterns of biodiversity and species interaction. Talk about cool science. I hope you're as excited as I am to welcome Natalie to Forest Talk. Natalie, it's all yours.
Okay, thanks for joining today, everyone. I appreciate being here. And I am happy to talk to you about one of my passions, which is Hawaiian spiders. So let's see. So you can see already from the intro slide that there's a huge diversity of like shapes and forms for Hawaiian spiders. And a lot of that is driven by their special ecologies. And so we're going to explore some of that today. I do want to give credit to George Roderick at UC Berkeley, who was my postdoctoral scholar advisor. And he took a lot of these, the majority of these photos are his. So we start by talking about the Hawaiian Islands and um, how it actually acts like a time machine for studying biodiversity. And the reason why is that you have this set of discrete islands, and then even within each island, you have these different substrate ages. And so, what that does is it sets up what we call a space for time substitution. And that means that we can look at these communities that are progressively older, and we can look for the species that have been around longer, that have had more time to evolve. And then we can expect that on the younger islands, there will be younger species. And this pattern is actually seen multiple times across different taxa, plants, and arthropods. And so you see kind of a hopscotch pattern where they move down the island chain from the older to younger islands. And islands really provide this wonderful system for studying evolution and ecology. And we can think of them again as being this very simplified system where as each island is formed, you have the effect of colonization and then species interactions, interactions with the environment as well as interactions with each other. And that leads to speciation. And there's also extinction events. And so all of this gives us kind of this ecological and evolutionary dynamics. And this is a lot of what I work on in the lab. And I'm trying to understand how that is being disrupted from the new species that are arriving, these sort of novel ecosystems that we're forming from non-native taxa. And this is a molecular phylogeny of spider taxa that were collected in Hawaii, and we got a DNA sequence for each of them. Now, you can probably squint and see that there are some OTU. A molecular operational taxonomic unit. And these, again, are molecular data, and what they represent is the relationship among the spider taxa. And each of these represents a unique clade, and it's a representation of the species. So this is actual molecular data that we got from sequencing spider DNA. And so it is a nice tool for me to show you the relationship between some of the major groups. These are families of spiders that are found here. This is Thomisidae. These are your crab spiders. They look very crab-like, and especially when you see them while they're moving around, they move side to side like crabs. We also have Philodromidae. Sometimes these are called the running crab spiders. Um, they are close relatives to one another, and they can be distinguished by like the length of this leg whenever you're looking at them for diagnostic characteristics. Then we also have the salticids. Salticids are jumping spiders. These are very charismatic. You may have seen videos of peacock spiders from Australia where they have these elaborate dances. You also might recognize this spider. This is a Hawaiian happy face spider. That spider is a part of Theridiidae, which are the cobweb spiders. This actually includes like your black widow spiders as well. On the Hawaiian Islands, there's a large diversity of Theridiidae, and many of them are yet to be described. In fact, we only recently discovered this individual right here, Rugothoides, with the strange triangle abdomen shape. But I'll be talking to you a little bit about some of the diversity in this group later on. The Araneae are orb weaving spiders. This is a spider that makes the classic circle orb web. And this genus itself, we're just starting to understand the diversity in as well. This is a linyphiid spider. These are sheet web spiders, so they build their web. It's horizontal to the ground. And this individual right here is actually a case of island gigantism. So when they got here, they got much bigger than the other individuals on the mainland. But again, with this group, we're finding all of this hidden diversity because many members of this family are actually really small and cryptic. And so we're only just now starting to get a handle on how many individual species that there are. And then the big family in pink is Tetranathidae. Tetranatha is a very large adaptive radiation on the island, and we're going to spend some time focusing on them next. Spider diversity really comes alive at night. If you want to appreciate the forest in a new way, the next time that you're on a hike, just stay out in the forest a little bit after dark, around 8 o'clock or so, and bring your headlamp. And as you walk around, you'll see that there are spiders literally dangling from the branches of the trees off of the leaves. They're there to be hunters in the nighttime, or they're waiting for prey to come by. There are a lot of these cursorial hunters or active hunters. This is an example of an Ariamnes spider, or nickname is a stick spider. This one has this cryptic against lichen. And again, we're going to talk a little bit more about this radiation in a couple of slides. So let's focus first on Tetranatha. And Tetranatha are a great representation of an adaptive radiation. Adaptive radiation is when you have a fairly rapid evolution of a diverse number of species that come from some kind of common ancestor. And that adaptation is a result of having different ecologies, so different ways of making a living. There are at least 60 species. Some of them, again, have not been formally described by science yet in this group. Different behaviors, shapes, sizes, everything. It's interesting because you actually do see some convergence in them. So there are repeated forms or ecomorphs in some of them where they always appear as green to match their background, maroon, brown, or they have convergence in their webs and their web shape, which leads to convergence in diets. And so we can actually plot this on top of the island chronosequence and see that we do see this progression rule where taxa are, the older taxa are found on the older islands, and then they hopscotch down. But importantly, each time that you get to a new island, you come up, the spider comes up with the same solution, okay? So this is kind of an amazing phenomenon, but these ecomorphs end up being represented over and over and over again. So if we start on Kauai, we see that there's this small brown, the maroon, and the green form. Now, you would think that the green ones, for example, when you get to Oahu, that, 2 and 6 would be each other's closest relatives, but that's not actually the case. They're each the closest relative to the ancestor that arrived on the island, and then they diversified in form each time that they got to, so here to Maui Nui and then to Hawaii Island. Each time that they arrived, they diversified again into this different ecology. And the ecology is related to the substrate that they're on, where they're going to be hunting and hiding from birds that are trying to predate them. You also see an interesting diversity in the diet that is associated with the webs. So the webs can actually be indicative of a species that can be very distinctive. You can measure the morphology of the web, like you might measure the morphology of anything by taking its measurements across. So how big is the web? How many radii does it have? How long is that the silk? If you were to do the silk all the way out, how long would it be? And you know, all of that is, of course, the capture area for the prey, and so that's going to affect what they can eat. And so this web structure has been compared to stable isotope signatures and molecular gut content analysis to see how that's driving diversification. But how do all of these spiders find each other in the forest? This was kind of a big question for a long time because they want to find their own species. They don't have super great eyesight. They're actually able to detect each other through sound and vibration, but that is only limited to a certain distance in the substrate, right? It gets muffled, just like your sound would get muffled when you're in the forest with the foliage and stuff. So we found out, and this was work that was done by Ashley Adams at UC Berkeley, was in my lab when I was a PhD student. We were in a lab together, and she did these mate choice trials. So she offered the male spider a choice of female that was his congener, or a female that was his conspecific, so same species, different species. And then she recorded how many times they went to the same species. And then she did it with extracts from the silk as well. And then she profiled what the silk extracts actually were, like what the chemical compounds were using a GC mass spec. And you can see that for the web builders and for the spiny leg clade, that there's a diversity in their chemical signatures. And so what she believes is that this is actually a part of incipient speciation for the spiders. This is a way that they can tell each other apart in the forest, and that helps to improve reproductive isolation so that they are only mating with each other, and it helps to kind of drive these ecological distinctions and reinforce that adaptive radiation. So it's pretty phenomenal and groundbreaking work that she's doing there. Now, it's not just Tetranatha that we see the repeated diversification of ecomorphs in. We also see this in the Hawaiian stick spiders. This is an Ariamne species. And again, it's this repeated diversification down the island chronosequence. So we can, correlate this. This is a phylogeny. So again, it's looking at the relationship among the taxa using molecular data and correlating it to the different species and morphologies that are found. We see three main different ecomorphs. There's an ecomorph that is this dark ecomorph and an ecomorph that is gold ecomorph. And then you also actually have this lichen ecomorph that I mentioned before, which is pretty cool, that allows them to blend into that. These little pop-out windows right here are showing you a close-up of what that color deposits look like, and how that helps them to become cryptic and camouflaged in their environment. Again, it's related to their ecology. So if we look at what the different species are and what habitat that they're found in, and what some of the morphology or shape is, they are these different colors, and they're in these different habitats that allow them to match to their habitat and be cryptic. And that's pretty amazing if you ask me. So how do spiders find each other? Well, we said it's chemical signatures. What happens when they do find each other? This is Tetranatha spiders that are actually called long-jawed orb weavers, is the nickname for Tetranatha. And you can see that they have very, very, very long mandibles. And then this is the fang that is down there. That's the fang that's kind of collapsed down. So those are the jaws. So what kind of prey are they eating that they need this big jaw? Why do they need those big fangs? Well, it turns out that it has nothing to do with prey capture so much as it has to do with finding a mate and doing the mating process. This bottom panel is showing you the pedipalp. That's the sperm transfer organ for the spider. And the female epigynum is in the center of her abdomen. So he walks around with his pedipalps and then he will insert that into the epigynum. And I have a cool video to show you what this looks like. This is from PBS Deeper Look. If you haven't checked out this series, it's a really great like vignette on natural history of different organisms. Uber Eats knows oh. You crave Crunchwrap Supremes. Sorry, I bet I got rid of the ads. So get a free Crunchwrap Supreme from Taco Bell on Uber Eats right now. Because crunch time. When football makes you hungry, get deals all season long on Uber Eats. Order now.
He locks his jaws onto hers to keep her deadly fangs away from him. This tiny hook latches onto her fangs on each jaw. They become a beautifully dangerous entanglement. Face to face, they curve towards each other. He raises his skinny pedipalp, ending in what looks like a boxing glove. Then he twists it in like a light bulb. Inside each boxing glove is his sperm. Once he connects, elastic membranes inflate and deflate, each movement propelling his sperm onward. Okay, this isn't exactly a quickie. After a few minutes, he moves on to the second boxing glove. It takes both alternating to deliver his goods. The whole time, he's still holding her back. His jaws, her jaws. He prepared for this moment earlier by collecting sperm from his abdomen with his gloves. When he's done, there's no time to cuddle, because if he sticks around for too long, he might still become her next meal. Sometimes love really is a battlefield.
Hi, this is Laura Clivens. You may have noticed a different voice on Deep Looked.
Okay, so I thought that video was just fantastic and kind of gives you an idea about the secret life of spiders. And this is happening in all spiders. It's the same mating mechanism. There's all kinds of cool stories that have emerged when people are getting into the behavioral ecology of this. And, you know, some of the female spiders are incredibly aggressive. And there's a story about a black widow species that actually the male does a somersault into the female and kind of puts the pedipalp in and then has to get out really quickly. And in that case, he only does one pedipalp and then tries to get away as fast as he can. And so they've evolved this somersaulting mechanism, which is pretty wild. So what's up with the color? Where do they get these different colors from, right? Well, there's these omochrome pigments that are laid down. It can be laid down in these layers, yellow, red, and black omochrome pigments. You can see that there's a green happy face spider over here. Why is it green? Well, that only has to do with what that spider has eaten recently and actually changed the color of the spider because they're quite translucent. So that's kind of a wild thing. But you also do see this incredible iridescence color. That is from guanine crystals. The thickness of the guanine crystals can actually be adjusted by the spider. It's a metabolic waste product, but it provides this reflective background and can make them appear really bright and iridescent. You do also see color polymorphism within individuals, so they can change color as they age and change color for crypsis. So this is Tetranatha hawaiiensis, where it can be green and match the green leaf background and brown and match the brown leaf background, and that typically does change with age. Tetranatha anui is named after the word for rainbow because you see all of this incredible color diversity in a single species, in a single population where they have this much variation, which is really remarkable. And then we have our happy face spiders. The happy face spiders are so iconic, and I think that they're also a good introduction if you have friends who maybe are not too excited about spiders and you want to say, hey, look, there's a fascinating spider over here. They're really quite small, so if you've looked for them and haven't been able to find them, they're easy to find, or not easy, but they can be easier to find if you look under the undersides of leaves if you're out during the daytime. They like broadleaf leaf plants, so ilex is a good one to look at them on. They're really only a few millimeters in length. They build small webs there, and they have this genetically inherited pattern of polymorphism. What's interesting is that a lot of the happy face spiders are actually yellow, so solid yellow, and they don't have any of the red or black deposited. And typically you see a ratio of about 2 to 1 for the solid yellow pattern to the colored patterns in a population. And it's thought, Dr. Rosemary Gillespie came up with this hypothesis that is being maintained by natural selection because it makes it harder for the search image of the bird that is trying to predate them. So they would get trained on one type of spider color. And then if you have kind of this mix of color spiders, then it makes it harder for them to find them and eat them. So that was her hypothesis anyways. Now these are also Theridian spiders. So this is like the cousin of the happy face spider. And a whole bunch of these are undescribed. A lot of diversity here that we just haven't had the time to do formal descriptions of. And they're just fascinating because, again, you see these color polymorphisms in them and quite a bit of variation within a single population. And what is the ecology of these taxa? They're also very, very, very small. And so, they really are kind of just understudy groups. Another group that has been a focus of mine is the Hawaiian cyclosa. Cyclosa are trash line spiders. This is a group that's found globally, and they're quite abundant and speciose globally. But then on Hawaii, these are all endemic taxa that we have been discovering. And they're called trash line spiders because they put in their web in the middle, a little bit of trash. Sometimes you also will see their egg sacs in that trash line, and that allows them to camouflage themselves again so that birds don't predate them. They're out in the middle of the day. They sit in the center of their web. If they're just the only thing in the center of the web, they're easier to pick off. And so the thought is if they fill up their web with trash lines, then they can hide a little bit better. Now, this is, again, a molecular phylogeny looking at some of the different taxa that we sequenced. This is a group that has not been worked on since Simone worked on them in 1900. And there's a student currently right now that's working on revising this group. There are an interesting selection of taxa that are morphologically distinct, and locally endemic, but are quite rare and have only been collected a few times. So here is one example. There are other groups that have different ecologies. So this is a species that CF means it looks like turbinata, but it's not turbinata, which is a mainland species, but it looks quite a bit like them. And you can see the really, really, really big trash line down here, very, very thick. This is a really distinctive spider that you might find on East Maui. There's also Cyclosa perkinsi. This one is quite widespread geographically, but it happens to be in a little bit more mesic or open and disturbed habitats. And then there's Cyclosa simplificata. This species is found across the archipelago. It does a kind of a loose web like this. Long bridge threads across big open gaps in vegetation. And then within that group, Simply Quicata, there's actually a nested clade, which I believe is an example of incipient speciation, where in this case, they always build their webs against the base of a tree. And so we've been calling them the tree species, but they're always in the depressions of trunks of acacia koa and ohia trees. There's another example of a radiation, but it's not adaptive, so it's not the ecology. This is a non-adaptive radiation. And these are those case of island gigantism. This is Orson Welles spiders. You can see my hand with a little meter stick. So this is quite a big spider web. You will find these fairly consistently in the native forest above in mid to high elevation. This spider is just kind of like really big and you know could be a little bit startling. They come out at night, so you can see them cruising around their web at nighttime. We actually did do a diet analysis because they have kleptoparasites. So these are spiders that make their living by hanging out on the web of other spiders. One of the kleptoparasites is Ariamnes. So Ariamnes, they are either free hunting or they can be facultatively kleptoparasitic. Another one of them is Argyrodes, which is a close relative of Ariamnes. So they just hang out in kind of this threads part of the web. They let the big Orson Welles be down on the underside of the sheet web. That's where he catches, or she catches a majority of her prey, and then they will kind of pick up the droppings. Now we're trying to track biodiversity in a very changing world. And so, you know, the question is, how is all of this going to be modified whenever we have taxa that are coming in, they're moving around through human activities. And we know that, you know, invasive species can cause an acceleration of biodiversity loss. We know that the major threat to biodiversity loss is habitat loss. And so how do we, you know, keep up with this? And one answer that we've come up with is by using some of these molecular methods. So a lot of that like species discovery that we're doing now with some of these really small cryptic spiders, we're doing that because we had a molecular signal that those species existed from doing some of this DNA barcode, getting these like short sequences of DNA for spiders that we collected. So we sampled across the Hawaiian chronosequence. This is work that I did for my PhD. At 14 sites across the Hawaiian archipelago, there were 6 plots per site. We were able to reconstruct alpha diversity and beta diversity at the site. We did this for all arthropods. And we were also able to do this per plant. So look at the association of each of the insects per the plant that they were collected on. That's what this orange graph is here. So it's a map of the associations. And the weight of the line is the number of taxa within that family. So you can see Tetranatha, they're a big group, right? And so they are, you know, found on nearly all of the plants that we collected. And that's because, as we saw, Tetranatha have these ecologies that have adapted them to these different substrates. We also can plot this over time. So this upper plot is showing the increasing alpha diversity over time. This is the substrate flow age. So this is our kind of proxy of space for time that I was mentioning in the beginning. And then we have our flow age. So these are our different volcanoes and the proportion of spiders that are found there. Now we do have some non-native spiders in there, and actually if we take that same graph and we overlay it with the non-native taxa, we can see that there are likely some disruptions that are happening, some novel interactions that are happening. And so our job now is to really go back and to try to interrogate this a little more. There's some ongoing work of doing some additional gut studies where we're using DNA to find out what the spiders are preying on and try to see how they're competing with the non-natives and sequencing the non-native guts as well, because we know that you can have a major disruption of the food web by having these introductions of non-native taxa. And so that is all that I have to share today. And I just want to say thank you, and I'm happy to take any questions.
Okay, well, first of all, I am in love with spiders. Oh my
Yay!
gosh. Yay is right. I mean, the whole time I'm like jawed, I have my long jawed, orb spider going. My jaw was dropped. I'm like taking copious notes and just loving this so very, very much. I have a ton of questions. Oh, I see some also coming in the Q&A. Let's start with Laura's. What new island forms do spiders evolve the same way each time, or does something new always show up?
Yeah, so a lot of the new island forms are, and speciation is related to their ecologies. So differences in the way that they do prey capture, and differences in the way that they're finding their mates. Those are the two that we kind of outlined today, and that's going to have a corresponding change in their morphology or physiology. So either a change in like the shape of their jaw for clasping their mate, the shape of the pedipalp, the shape of the epigenum. So those are like a lock and key mechanism, and that's the main way that you can tell spider species apart, is by looking for that lock and key mechanism. But you're also going to see adaptations in their camouflage, right? Their ability to be cryptic, and these color polymorphisms that are thought to help them to avoid prey as well.
Just fabulous. I'm like still loving all of this. Okay, Jerry has a question. How do spiders get from one island to the next? And is that still happening now?
That is a great question. So spiders, a lot of spiders do something called ballooning. Not all groups, but a lot of them do something called ballooning. If you are a fan of Charlotte's Web, I was a huge fan of Charlotte's Web. So in Charlotte's Web, they describe ballooning, where the spider will go to the a top of a fence post or a tree or someplace that's a little bit higher up, as a juvenile. So when they first emerge from the egg sac, and then they're going to disperse away from like their natal range, and that's to evolutionarily to keep from like mating with your closest relatives. And they shoot a little piece of silk off of from their spinnerets. That piece of silk acts like a balloon, and it can carry them away. And so the spiders that were colonizing each of the islands, they were doing that probably through ballooning. They also could do that through rafting. So if they're caught up in any type of debris during a storm, they could raft over between the islands. But as you can imagine, these long distance dispersal events to the Hawaiian archipelago are quite rare. And so there's very low colonization from the mainland, and probably not a huge colonization between the islands. And so that's why we see most evolution happening within the island. And those are each other's closest relatives. And that's why it's a really curious thing that they came up with these same ecological strategies, and these ecomorphs every time that they got to a new island.
It's just fascinating. I even love the terms of their anatomy that you're tossing out. Spinnerets and the
Yes.
pedipads, is it?
Pedipalps.
Pedipalps. I mean, I'm loving this. And you mentioned Charlotte's Web. How did you get into spiders? How did you find this to be your life's work, if you don't mind sharing?
Yeah, well, I grew up in the country, and I spent a lot of time outside as a kid, and I was the kid that like pokes the proverbial ant's nest. Like I was always just kind of like playing outside in the grass and the trees and stuff, and looking at things. And so I think that's part of like why I wanted to become a biologist. But then later on, when I got into like my undergrad and grad school, I started thinking about what questions excited me, and I was really interested in how biodiversity is generated and maintained, which is the process of speciation. And I got super into this idea of being able to use DNA tools to tell taxa apart and to look for kind of these hidden lineages. And then when I did my PhD, I was in a spider biologist lab. She's quite well known, Rosemary Gillespie. She's the one who described the Ariamnes species and the Tetranatha species and a whole slew of other spiders on the Hawaiian Islands. And so she asked if I would teach the corresponding laboratory for her spider biology class. And so I taught that for a few years, and that gave me a huge love of spiders and spider ecology. And so, yeah, that's my story.
Well, I love it. And had I been, hanging around her and or you when I was young, I might be in the spider lab too. I might retire and decide to join the spider lab. It's so fascinating to me for some reason. Um, but so let me, and what I love too about what you just described is how once again we have a species that describe it talks about what makes these Hawaiian Islands so
Yeah,
unique, the adaptive radiation. We see adaptive radiation in plant species, in bird species. The most famous one, of course, is the honey creepers, which
mhm,
supposedly, we think a single rose finch species
right.
flew in, maybe in a flock, and then over so many generations adapted to the unique microclimates of each different island, and we're seeing that in the spiders too, to have 50 some different species of honey creepers that we know of, maybe more, who knows. So we see it in plants, we see it in spiders now, we see it in all kinds of insects too. So anyway, I love it. Thank you so very much. So quick question. Um, do we know how many spiders? I know that not all of them have been described. How many spiders native in Hawaii at this point in time estimate, would you say?
Mm.
Sounds like a lot.
Yeah, I'm trying to think. So some of the radiations, Tetranaba is the largest radiation uh, to date, which is 60 plus species. But if we go back to even just that first phylogeny, right? So all of these families are represented here. Now, I should also say that there's a whole bunch of spider families that are not on Hawaii. They never made it here. They didn't win
Sure.
that sweepstakes lottery of long distance colonization, and so they don't have any diversification represented. But there are like Mecaphysa is a radiation in the Timicids. There's Philodromidae. There's several species there. So, even if we, said that there's, 30 species in these other groups conservatively, then, you're looking at several hundred spiders that
Amazing.
are
Yeah.
distinct. Yeah.
Yeah. I
And
love
I
it.
think that's a conservative estimate because there's a lot of these groups that we're just starting to discover. A lot
Which
of them
is,
are these single island endemics. You're only finding them on one island or one mountain.
yeah. Yeah. Which, on one hand, wow, so much we don't know, but that is to me such an opportunity for young people. So I'm always trying to like encourage we have friends who have children who are like pondering their careers, be it, children, grandchildren, friends of nieces, nephews. I mean, here's like there's a place here. We need people to
Yes.
come in and help us with this because there's fascinating and B, a lot to be done and a lot
Yes.
to be learned. Yeah. So it sounds like too, you touched on basically endemism that we have, these species that are found in Hawaii and nowhere else in the world due to speciation and potentially in specific islands and mountain tops or certain microclimates. Would you say the vast majority of our native spiders are endemic?
Yes.
Yes. Okay.
Yeah, so very much so. And actually the majority of spiders that are here are native or endemic, and there hasn't been a huge amount of infiltration of non-native spiders into the forest. Now I showed you some of the ones that are getting in there. So Steatota, which is the false brown widow, and is another cobweb spider. Chiracanthium, which actually has a really nasty bite, and it's a big pest in Europe. Um, and that's kind of an interesting hypothesis because there's some thought that because they're so diverse in the ecological niche space that they've filled already, then there's not a lot of room for non-natives to make it into our nice intact Hawaiian forest. But as we modify the habitat and bring in invasive plants, we're making room for some of these non-native species to come in, and we've been seeing a new non-native Badumna that we're now finding it at Hakalau, which was not there, before,
Hmm.
even 5 years ago. So there's starting to be encroachment, still mostly around human habitations, roads, edge effects, but yeah. So we want to keep those out as much as possible.
Yes, yes, for sure. Yes. And hopefully that's where the ISCs might come in, our invasive species committees across the islands. Tammy is asking, has two great questions. Are spiders ambidextrous? First question. And second one is, are there venomous native spiders?
Okay, those are two good questions. So yes, ambidextrous, and they have antennae, and they have their pedipalps, and they have all these like sensory organs, hairs and spines and things like that. So they're quite adept at moving around and in the environment. And yes, they're so fully ambidextrous. And then the second question is, oh, sorry, remind me of the second question. I
Sure.
just
It's, are there venomous native
Yes, okay.
spiders?
So all spiders are venomous, and like universally. And then it's just that the venom is not tuned into to humans. So it's just targeted for their prey. Oftentimes they don't have fangs that can really hurt us. I mentioned the Chiracanthium spider species that is sort of plaguing Europe and is now here. So that's a case of a spider that can do a bite. But in these cases, you could let them walk all over you and they wouldn't hurt you. It's just the non-native ones I wouldn't do that with. The native ones, no problem.
So that kind of leads me to a really far out maybe question. But with science in nature, we do see some bizarre things that happen. Um, and it sounds like what I've always understood is that spiders prey on insects primarily, maybe even other spiders, as you've talked about. Oh, and I love that we have some kleptoparasitic spiders.
Yeah.
Thank you, because we have those in birds too, right? I'm like, oh, in
Yeah.
spiders, of course. But is there any science that shows that spiders are using plants as a food source itself?
No, they're strictly predatory, but they can prey on, they've been known to prey on vertebrate species. They feed on a variety of arthropods, so they can feed on like the isopods and like other non-insect arthropods as well.
Okay.
But yeah, totally predatory, and that's a part of the way that they like digest their food and stuff as well.
Okay. All right. That
Yeah,
would be too big of a jump. Yeah.
yeah, yeah.
Um, oh, Volker wants to know, are native spiders found in lower elevation among largely invasive plants, or are they restricted to mostly native forests higher up?
Yeah, that's a good question too. So, there are pockets of low elevation forests that they are found in, like the Blue Hole area on Kauai is a good example. And there's some other areas that are, more like 900 meters, 800 meters, where you still see forest that is maybe somewhat introduced species, but it was probably remnant forest that was at kind of lower elevation. The majority of the spiders that you see in like around Hilo Town are not native.
Mhm.
And Yeah, you have to go, mostly you have to go into the mid to high elevation forest to find native spiders, with some few exceptions.
Yeah. And back to, our everybody loves the happy face spider. Um, has that been confirmed on Kauai? I hear conflicting. Do you know?
Yes. Yes. So
We have this
um,
one thing that we
I
call
don't
the
have a,
spooky face spider. Is that just a
yeah.
different color morph?
Yeah, I guess I don't have the phylogeny of them. I believe that they're on every island. Um, I believe that they're on every island. Yeah.
Okay.
Um, but I could be wrong.
Well, we'll all have to look for them here, those of us that are in the room from Kauai, and we will definitely report back.
Yes.
In general, Jerry asks, how do we help protect spiders across the islands?
Yeah, that's great. Thanks for that question, Jerry. I think that the main thing is to preserve the habitat, right? So, and they're hanging on in surprising places. So if you know Telephone Pole Road on Tantalus, that some of that area is like bamboo. There's a whole bunch of houses up there. But we can consistently go up there and find like 5 or 6 families with like multiple species represented. Even to the point where we were like, we have to stop collecting here because this is probably just this tiny little holdout area. So, protecting the habitat is a main thing, and they are tuned into the plants as well. So, with insects, the number one thing is having the native plants. They need the native plants to complete their life cycle. And there's some of that same evidence in spiders, right? They're matched to their substrate. They're have these hunting habitat habits and all of this like diversity that's tied to their the native forest, high or low elevation. And so just trying to maintain native plants and recruit native insects is the best thing.
You know,
Don't spray a
that's
bunch of stuff. If their pesticides are bad for our native spiders also. That's another one.
Yeah, it's what we talk about with ohia so often, right? Is, to save all these other plants and animals, we do need to ensure that we have a healthy ohia forest, ohia as the backbone of our forest, and providing so many, services, ecosystem services, in addition to, the water that we drink. Um, and the thing that I often talk about is biosecurity and, specifically how not to spread rapid ohia death. And we talk about making sure you're not moving mud, or other pathogens around the island or from one hiking trail to another. And in addition to helping save ohia, that will help prevent these other perhaps invasive spiders from getting in or other invasive plants, from taking over the habitat. So, it's that becomes a universal message. If we want to save and protect pretty much anything on these islands, it's about habitat and it's about biosecurity. Okay, I think last question, and then we'll let you go. This has been
This is great.
so much fun, I have to tell you. When can I buy your upcoming book, The Complete Guide to Hawaii Spiders? Yes, great
So
question.
that's funny that you should ask that. So Dr. Gillespie, Rosemary Gillespie, is working on a book that will be a bit of a field guide and a description of all of the taxa, their special ecologies, lots and lots of pictures. And so she's actually been working on this. And so I will report back that there is interest in this book because we've been having some conversations about like who will buy it, and I've said, well, I'll have my students buy it, and so I think okay, good.
No, I love it. Thank you for that question. Yeah, one of the things I'm thinking is, oh my gosh, I want more information. Where can I get more
Yeah,
information? And so, yes, the book would be, I think, a big hit.
excellent.
And I just think it's fascinating. And I'm so thankful that you're willing to share your knowledge, just to create more awareness for yet another amazing creature in our native forests, that we want to learn about in order to protect it. And unless you have any last comments, I'll give you the last opportunity if you want to maybe share the website for your lab.
Oh yeah, I can put the website in the chat. We're just ilalalab.org. And that has my research group and my students and all of our activities working with community ecology and invasive species stuff. So I'm super excited to talk to anybody about Hawaiian spiders ever. So
Well, when you get back over to Kauai, please reach out if you have any time. Would love
will
to meet
do.
you in the forest and talk more. And for the rest of you, again, thank you for showing up. When we do close this out, as always, you're going to have that pop-up screen that comes up on a new tab with just, I think it's 3 short questions about today that helps us with planning for future Forest Talk sessions. And I hope everybody has a Halloween costume in mind that they're going to rush out and create right now. And you've been inspired by this. So thanks, everybody. Have a great day. Thanks,
Thank
Natalie.
you.
Aloha. Ahuyo.